Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the Associate Editor-in-Chief of Molecular Therapy Advances, Dr. Daniel Stone, and Dr. Krishanu Saha, Anna Tommasi, and Dan Cappabianca from the University of Wisconsin-Madison. They will discuss an article recently published in Molecular Therapy Advances by Tommasi and colleagues titled Efficient Nonviral Integration of Large Transgenes into Human T Cells Using Cas9-CLIPT. Before we dive into this breakthrough in nonviral integration, I want to invite you to ASGCT's Empowering Patients Summit on March 11th and 12th. This free virtual event bridges the gap between the lab and the community, bringing together researchers, advocates, and industry leaders. You'll gain direct access to experts and see how patient-driven insights are shaping the future of cell and gene therapies. Learn more at asgct.org/empowering. And now, Dr. Stone, Saha, Tommasi, and Cappabianca.
Okay, well, hello and welcome to the Molecular Therapy Podcast. My name is Daniel Stone. I'm your host for this episode, and I am the Associate Editor-in-Chief of the ASGCT journal Molecular Therapy Advances, formerly Molecular Therapy Methods in Clinical Development. Now, today we will be discussing a paper that was published last year in Molecular Therapy Methods in Clinical Development titled Efficient Nonviral Integration of Large Transgenes into Human T Cells Using Cas9-CLIPT, which describes a new CRISPR-based system that can genetically modify and edit human T cells with efficiencies of up to 60% on a scale that permits clinically relevant yields. Now, as we move into an era of personalized medicines, methods to simplify donor template production and integrate large transgenes will allow for the improved manufacturing of multifunctional genome-edited immune cell therapies, and the Cas9-CLIPT method offers a way to do this more affordably and at a clinical scale. Now, the Cas9-CLIPT system was developed in the lab of Dr. Krishanu Saha from the University of Wisconsin-Madison and the Wisconsin Institute of Discovery. And today I will be joined by Chris and first authors Anna Tommasi and Dan Cappabianca. It's my pleasure to introduce them to our audience. So Chris received his bachelor's in chemical engineering from Cornell, his PhD in chemical engineering from UC Berkeley, before doing a postdoc under Rudy Yanish at the Whitehead Institute for Biomedical Research at MIT. Chris is a professor of biomedical engineering, pediatrics, and bioethics at the University of Wisconsin-Madison. He holds the Retina Research Foundation Catherine and Latimer Murphy Chair at the McPherson Eye Research Institute. Chris and I both trained under Dave Schaefer at UC Berkeley, although Chris graduated and joined the Yanish lab a few months before I joined Dave's lab back in 2007, and we only met for the first time last year at the SGCT meeting. Very good to talk to you today, Chris. Now, Anna received her bachelor's in chemical engineering from Villanova University and is currently a Saha lab member and a graduate student from the UW Biomedical Engineering program. Dan received his bachelor's in chemical engineering and biochemistry at the University of Wisconsin-Madison and is a former Saha lab member, earning his PhD from the UW Biomedical Engineering program. He's currently a postdoc in the SOGI lab at the University of Wisconsin-Madison. Chris, Anna, and Dan, it's a pleasure to have you with us today. Now, I thought I'd get us started by asking you something, Chris. Chris, I was wondering if you could tell us a little bit about the focus of your lab's research and what was your purpose for initiating the work that we're going to talk about today?
Yeah, thanks for the question and great to see you, Dan. Thanks for the invitation. It's a pleasure to be here. Yeah, I'm happy to expand about our ex vivo cell therapy projects. We're very interested in being able to program human cells ex vivo, and a lot of that work originally started with human stem cells, so embryonic stem cells, pluripotent stem cells, and we've been excited by the prospect of engineering immune cells, both mature primary T cells as well as primary NK cells. And so that work has been supported generously by the National Science Foundation, as well as the National Institutes for Health. We've been working on a strategy for about a decade now, trying to knock in long transgenes, namely the chimeric antigen receptor, which is obviously clinically relevant now with several approved products, without having to use traditional viral vectors, be it retroviruses, lentiviruses, or other strategies, and to try to do that fully with as well-defined kind of synthetic reagents as possible. And so that work, really pioneered by two former graduate students, Nicole Piscopo and Katie Mueller, has now led up to this publication that describes this new strategy to make the donor template in a virus-free way. So this is a large collaborative project. We're interested in working and partnering with industry. In this case, it was Aldevron, and you'll hear about some of our more translational work with some manufacturing partners such as Solaris. But the goal here is to be able to quickly come up with a transgene design and make a batch of gene-edited, gene-engineered cells that would be therapeutic. And so trying to reduce that time as much as possible and to make it as arguably as affordable as possible to explore a lot of designs is where this work has been going.
Okay. Well, that's great. Thank you for the kind of overview of the lab. So I'm going to kind of just to give a little bit of background on the project. It's probably better if it comes from one of you guys. So Dan, I was wondering if you could maybe give us an executive summary of the Cas9-CLIPT system before I kind of dive into some more detailed questions.
Sure, I can definitely do that. So I kind of helped start this project with Anna and one of my talented undergraduates, Madison, when I was in grad school here. Before we probably go any further, I should probably say what the Cas9-CLIPT platform stands for. The acronym stands for Cleaved Linearized with Protein Template. So effectively what this means is that we have, when you're doing gene editing with CRISPR-Cas9, you have to have something called a homology-directed repair template, which is a piece of DNA that's going to be encoding the gene of choice. For us, that's, like Chris said, a chimeric antigen receptor. Sometimes you can add other things to this to help the CAR function better in vivo, i.e. armored CAR T cells, which is the interest in inserting longer transgenes with this method. So what basically happens is you include this little sequence on there that usually the Cas9 recognizes this guide sequence plus a PAM sequence inside the gene itself, which allows it to make a double strand break so that the endogenous repair enzymes can insert the transgene in. We put this on the plasmid itself so that it linearized, as a homology-directed repair template needs to be linearized to actually insert. And this, surprisingly, it helped overcome a lot of challenges in making the template. We had a lot of manufacturing problems, probably not problems at the start, but it was inefficient at the start to make this kind of template, because typically groups like the Marson Lab, other groups out of UCSF and Minnesota and even China have done work where they amplify a regional plasmid, and then you have to purify the DNA from there. We simplified this by making the system such that you can just order this from the company Aldevron, their nanoplasmid platform, with your transgene encoded, and then we just add the Cas9 in when we're electroprinting our cells, and it's a one-step process.
Well, that sounds really simple, relatively simple, as you know, things are often not as simple as you think they are. So what prompted you to use the nanoplasmid system rather than the more PCR-based, a PCR-based approach? I know other people have been using the mini-circle plasmid system. Was there anything in particular that drove you down this pathway?
I can bump that over to Chris. I think you have a talent for seeing things in industry that really, you brought that to the table.
I think the strategy likely could work with many types of plasmids and kind of small backbones. The advantage of the nanoplasmid from our point of view has been that it has some background in being used clinically, not necessarily as a homology-directed template, but for expressing other transgenes. But the principle that I think we describe well in the paper is this addition of arguably a very short sequence, 20 to 23 base pairs or so, that confers a target site in the template that really changes topology. And so that is not necessarily limited to the nanoplasmid offering from Aldebaran, but really to, I think, other types of double-stranded DNA molecules. It could be others with different backbones as well, we think could also benefit from having Cas9-1 cleave and then likely associate and stay, at least for a period of time, associated with that template. And that's where the protein terminology in CLIPT comes from. But of course, we are very excited to move forward with the nanoplasmid strategy. And I think, Anna, you can say more about our work with nanoplasmid and how we're applying it to a particular case of anti-GD2 CAR T cells.
Yeah, it's a really exciting project we have going on right now. Like Dan was saying, just at the benchtop scale, it took far too long and way too many materials that were quite expensive to try and linearize with a PCR-based assay our donor DNA when trying to perform these experiments at scale. And so this Cas9-CLIPT technology that we developed has really helped scale up our donor DNA manufacturing, basically. And so it kind of removes this whole week of work that we used to have to do in order to make enough DNA to just do a simple scale up of our experiments. And so it's just translated really well to these at-scale experiments that we're running with our third-party company collaborator, Solaris. They have kind of a one-stop shop electroporator device, because that's also how we make our CAR T cells, is with electroporation. And so it makes it really simple. All we have to do is put our nanoplasmid, our Cas9, and our guide into their electroporator with the T cells, and we get CAR T cells out of the process instead of having to do this PCR-based linearization prior to adding our donor DNA to the T cells.
I'll add that in terms of quality control and the requirements from the FDA is much easier to do sequence verification on a master cell bank that's making the nanoplasmid than on every batch of PCR amplification. And so in that way, Anna said it was quite easy and simple. It's actually been a year-long process to get this up to scale with the right type of quality attributes that would be appropriate for an IND filing to the FDA.
No, that's really interesting you brought those points up, and I have some questions a little bit later on that kind of get to the scalability issues. But before we kind of jump into that, being more of a molecular biologist by trade, I have some more kind of like details questions for you. That's kind of the way I think. So I was, you obviously compared the CLIPT system to, linearized plasmid and some PCR templates, and it seems to be performing better. Why do you think that is? In theory, you have the same linearized template there. Really, the main difference is you've got the bound RNP once it's electroporated. I'm not sure who the best person to ask that is, but I'll throw that over to you as the whole.
I can take it.
Sure. Why don't we go ahead, Dan?
Yeah. So I think it's actually kind of a simple mechanism where the Cas9 has what's called an NLS, or a nuclear localization signal, on it. So when the DNA is bound to it via this like Cas9 basically shuttle sequence, it basically just takes more of the DNA with it to the nucleus when it goes.
So that's okay. So you think it's as simple as that? Okay. So then
one way.
So I have a follow-up question to that then, and this is related to Chris and I's somewhat shared past, even though we didn't work on the same thing in the Schaefer lab. So as you guys know, AAV is being widely used as a donor for, very similar approaches, albeit with a single-stranded DNA template. AAV can go to the nucleus. So how do the editing frequencies of this system compare to what other people have seen with AAV? I know you don't compare the two here head to head, but, just kind of on a rough idea, do you think this is obviously much easier to develop, than growing an AAV, making AAV donors, et cetera. But how do they kind of compare?
Yeah, funny you asked that question. I was just talking with a colleague here at Madison about that exact question. As you said, we didn't do a side-by-side comparison. I think generally we don't expect it to perform up to the levels of having AV6, for instance, as a donor template, which I think I've seen some published data which gets into 80 to 90% plus knock-in. So we're at 60% knock-in for the templates that were for the CAR, a bit lower for larger templates when we add a template up to 5 kb. So I think overall non-viral strategies in our programs outside of T cells as well for the in vivo programs that we have in the lab are just generally lower compared to viral strategies. We do think 60%, anywhere in the range of actually 15 to 60% seems reasonable for the type of ex vivo strategies that we're going for translation. I think there's potentially another layer to the explanation that Dan mentioned, in that the innate response of T cells to double-stranded DNA may be different when we have some protein on there. And so that could potentially provide a survival advantage or cause apoptosis in some of the cells that are either edited or not edited, or naive or effector. We didn't really go into that.
Have you tried any other kind of additional drugs or like similar approaches to kind of bias this, or and how, and also, is this something that's amenable to clinical scale up? Is this kind of are these acceptable drugs to be using for the FDA for these kind of approaches?
I think, Dan, you can talk about the array of drugs you've tested. In general, I think these are a class of DNA repair inhibitors that either block non-homologous end joining. Some of them have been used clinically at various types of doses. This is ex vivo application to basically cultures where that factor would be diluted away before infusion, or it should not really persist in the cell eventual product, where after electroporation, there's arguably 3 to 7 to 10 days of subsequent culture where it gets diluted out. Yeah. But maybe you can go into all the different types of molecules you tried.
These are kind of the only two we actually did try. But they worked. They were the best ones from screens of other papers, which is why they were good candidates. So the nadisertib it's a protein DNA PK inhibitor, which is, like Chris said, a protein that's very involved in non-homologous end joining. And then the ART558 it's a polymerase theta inhibitor, which is more like MMEJ, HMEJ. It's a whole bunch of names for that kind of in-between repair mechanism, I like to say. But I think the nadisertib was the one that kind of caught our eye the most, because that's the one that's more involved in clinical trials where they combine it with like chemotherapy drugs or radiation, where it helps sensitize cancer cells to either like ionizing radiation or just helps stall their DNA damage response. In this case, it just helps boost like a competing DNA repair pathway, homology-directed repair, and just consistently increases the knock-in of the cells by about twofold, I'd say. So because it's so involved in a lot of these clinical trials, we just thought it was easy to slip in as an ancillary product to a lot of these things. But Anna can kind of talk more about anything clinical.
Yeah, well, actually, that's another follow-up question, which may be appropriate for Anna here as well, because you're also using PGA for different reasons, obviously, than to kind of bias the HDR. Again, is this something that would be acceptable in a clinical product in terms of scale up? You kind of, some of your results that seem to help, some of your conditions that seem to help much more than others. And, it definitely seems that there's, logic for using it. But is this something that is applicable to something like that, Hannah?
Yeah, great question. Just a little bit of an add-on to what Dan was just saying. We did try a few other inhibitors like an AZD7648, but didn't see much, which is also a DNA PK inhibitor, but didn't see much of a response with that, and also tried an HDR enhancer. But again, those two didn't really improve our system at all. And so we kind of just moved forward with the M3814 and the ART558. But correct. At clinical scale, a lot of these small molecules aren't GMP compatible or certified. And so it's been difficult to try and integrate them into our system with Solaris. And we're finding that we have removed a lot of those small molecules and PGA from the manufacturing process at clinical scale, just to make sure that we are still complying with all the FDA standards and making sure that our process still works on Solaris's machine, because translating this manufacturing process from one electroporator to another, you get varying results every single time. And so we have just found that some of the small molecules and inhibitors that we used on our benchtop scale instruments in lab here don't really translate well at the clinical scale.
Yeah, I'll add that some of these pathways of blocking end joining or microhomology-mediated end joining may be dependent on the exact sequences in your donor as well as in the resected double-strand break, and then also perhaps dependent on the electroporation system. And so for our kind of project that's in translation, which is a GD2 CAR, we found that you can remove some of these components that may be harder to obtain at the sufficient quality as well. So, I'm a proponent of simpler is better, and in some ways, this could really, these molecules can help in one setting quite a bit. That's why we wanted to make sure we included that data, but may not be essential.
Yeah, no, really good points there. And I definitely second your simpler is better. So before I get into that a little bit more with the expansion, just one kind of follow-up question in terms of the constructs you've been using. So in your paper, the largest insert I think you have is about 5 kb there. And, most of your work is with that, or your 2 kb construct. Have you tried anything larger yet? And have you also kind of tested this system at other safe harbor sites, such as AAVS1, or, in other systems like mouse systems, Rosa Locus, or anything like that? Is this something you're considering?
Yes, we've done some pilot experiments with larger templates. I think the largest one we've knocked in at lower rates, but successfully, is around a 7 kb transgene. And then in terms of other spots in the genome for integration, yes, AVS1 we've been able to integrate into as well. We have not done work with mouse cells with this strategy yet.
Sure. Obviously some people just go straight to human, and that makes perfect sense. Okay. So let's move on a little bit to the manufacturing side of it then. So you show some data in the paper where you've used the Lonza nuclear effector, which probably a lot of people would be more familiar with that system. You mentioned the Solaris system, which I was not familiar with until I'd read your paper. And then you mentioned the Neon system, which some people might be familiar with, which is kind of translatable to the larger scale-up system that Thermo have. And you obviously, it's different donors, variability, you get variability from system to system as we talked about. But obviously works better in the Lonza system. But this is more from my perspective. I'm not really familiar with this. Which of these systems would you say is more scalable? And do you think there's room for further optimization within these different platforms?
I can start, and I think, Anna, you can share more about the experiments in the lab. I think all of them are scalable. There's different instruments that need to be used to get up to sufficient higher volume electroporation. So the neon has a kind of sister piece of equipment called a xenon, and both of those we've been working on together that seem to work well. The Lonza system also seems to work well too. The parameter systems are hard to translate from one instrument to another, and that's a whole set of, I think, physics and biology that we're still fully trying to understand. Much of this is empirical testing from donor to donor with construct to construct. The Solaris electroporator really is available through Solaris, and they've been very generous in working with us and helping us with that process development. I think we are one of the first to work with them to try a virus-free protocol, and that so far does seem scalable. We are still in the middle of that process development that Anna can tell you more about. But I don't think the kind of biology that we described and the system we described in this paper is necessarily tied to one electroporation system.
Yeah, I think I would agree with that. Kind of the story behind using all the different electroporators was the work that was done prior to me and Dan working on this Cas9-CLIPT paper that Chris mentioned earlier with Nicole and Katie Mueller. They used the Lonza system in their paper. And so that's kind of just what we had in lab. And then as a campus, UW got this xenon electroporator, which is kind of a large scale electroporation device. And so trying to translate our Lonza protocol to this now large scale electroporation system proved to be a little bit difficult because just the scale of materials that you needed in order to run these like large scale experiments on xenon was kind of crazy to just do these like pilot experiments on, and so we acquired neon, which is the small scale version of xenon, to do all these translational experiments from Lonza and making sure our process still works on neon so that we can then scale up to xenon, kind of with the idea that in the future we were going to work with Solaris and try and get this therapy into the clinic. And so that was the goal behind the large scale xenon experiments, and then running all these experiments on other electroporation devices as well.
Yeah. And I know that in our group, we've had some, we've played around with the neon and the AMAXA in terms of transducing T cells for HIV studies. And we see some problems going from one to the other, more so because of the buffers that you don't know what are in them that are all confidential, and that kind of definitely can make it a little bit challenging. I wonder if you've had similar kind of issues there.
Yes, big time.
There's quite a large parameter space, and I think one of the challenges and why it seems crazy to try to optimize a parameter space at the large volume is that it is quite expensive, and that's why there's these smaller scale systems that, in theory should be entirely scalable to the larger system. But the geometry from a 1 mil cuvette to a 20 mil kind of flow system or system with tubing that has different dead volumes, etc., it does make it challenging for really any of these instruments. And so I think coming up with better delivery strategies, even different instrumentation with electroporation, could really help increase efficiencies and scalability here. It's not as in some ways sexy as coming up with a new delivery vehicle for in vivo transduction, but very important, could help ex vivo manufacturing quite a bit.
It's a really good point. And so, kind of following on for the manufacturing side of it, so in some of your phenotypical analysis of the products you're getting out this, you had a really nice mix of CD4 and CD8 positive modified cells, and then a good mixture of, like the different naive central memory effectors, etc., populations in there. Were you happy with this? Although I work at Fred Hutch, I'm not a CAR T person, so I couldn't tell you whether these populations would be expected or what you'd want. Are you happy with these products? Are they comparable to, let's say, Lenti products that other people are using? And I know the proof is in, testing it in animals or whatever, ideally a lot of people, but, from a phenotypical perspective, everything's looking how you would like it to be.
Yes, I think the short answer is yes. I mean, we haven't done the kind of long-term in vivo experiments in mouse studies in this paper, but our previous paper did some in vivo studies, and it matches those profiles in terms of the types of memory markers and effector markers. It's in the range of what we've seen many others, even with clinical products, have in their products. So yeah, I mean, we're not concerned. We didn't see, huge levels of exhaustion or really any other markers that would give us concern.
Great. Well, that's really nice to hear. Okay, so and then as I guess a little very quick follow on from that, have you tried any other tumor antigens except for the GDT antigen, or for now you're just kind of working on this and then you expand it later on as needed, or do you have any other kind of target cancers that you're looking at and antigens?
Yeah, we have a preprint that was released with UPAR as one of our CAR targets, and that's a senolytic strategy. That's a really new story that we're trying to tie up. But in terms of other targets, CD19 and B7H3, those are all different targets that are really exciting, I think, as directions that we could go. We haven't published any stories on there, but we do think this strategy could work there as well.
Great. And then another question I have regarding, quality control of the products. You've very well characterized the on-target and the off-target editing, via nanopore sequencing or whole genome sequencing. And you've got some really nice on-target integration rates there. But I was kind of very interested in one of the comments that you'd put in the discussion about integration into chromosomes 2 and 22, possibly being due to immunoglobulin genes and homology to the single chain Fe region in your CAR. I know in some of our work, when we've been doing viral mediated studies, we kind of see some low level off target effects when you have like a sequence that's present elsewhere in the genome. So I was wondering if you actually sequenced around these loci to see if you had actually like some kind of tethering effect there, or this was just kind of a response to a reviewer comment.
I'll start, and then Anna should talk about the experiments. I believe that much of the match to those regions was due to our transgene sequence being basically borrowed from those endogenous sequences. There is a limit of resolution of our assay in terms of how precise we could see off-target integration. And we did not go in and further investigate by amplifying those additional regions that you discussed, Dan. But I do think, this was actually our first work in using whole genome sequencing analysis to look at integration. Part of this was inspired through some work in our NIH consortium on somatic cell genome editing, where they have a number of projects that look at integration and off-target effects from genome editing. But this was actually a really exciting direction that we developed with this work. Anna, maybe you can talk about the algorithm and how you developed it.
So we have this postdoc, or had this postdoc in our lab, Kirsten Gimpsey, who definitely knows a lot more about the bioinformatics side of things. And so she developed this program through Minimap2 that was able to map our CAR transgene and everything against the whole genome of a human. It was really, really fascinating how she was able to do that. But yeah, she did get these hits for these off-target sites. And like Chris was saying, we just attributed those to the single chain variable fragments, I guess, that you kind of see in our CAR and also in chromosome 2 and 22. So we didn't see too much of a cause for concern, especially considering there were such low numbers of off-target hits at any of these sites. It would be really interesting to try and do some more targeted sequencing around those to confirm, but we are certainly pursuing a lot more sequencing and genotyping and all that cytogenetic also sequencing for these clinical scale experiments that we're running. So hopefully soon maybe we get some more information about the genetic impact of all these edits.
Yeah, I mean, as a group that uses AAV, we're obviously seeing probably a little bit more off-target issues for due to that. We've kind of trying an approach where we do like a probe capture based study. So we find that it works quite nicely for unbiased sequencing. There's maybe a little bit more, you probably get a little bit more coverage than the whole genome sequencing approach. So you might want to kind of consider something like that. But it's really nice to see this whole genome data because it's not easy and it's getting better, right? So hopefully it'll be able to pick up even more and more of these events that are happening at low frequency so we can really get a good idea of the big picture.
Yeah, no, I absolutely right. I mean, the cost of whole genome sequencing is really dropping, and that's where at a limit of resolution to make sure that we have at least enough ratio of on-target to, off-target events is super important. And these reads that we saw were pretty phenomenal from a nanopore, up to 30 kb reads.
Oh, wow.
That really helps you say, okay, we actually had one read that had the entire transgene at the exact spot with the right homology arms. Gave us really, I think, better confidence that we are getting homology-directed repair.
Yeah, no, very important to follow up on that, and that's really, really great data. So kind of coming close to wrapping up here, I think, but I wanted to ask you just a couple more questions before we go. And they're kind of more big picture, so maybe we can kind of start with Dan on this one and see if anyone else has something to add in. So obviously the main advantage of the system, I would say, would be affordability, scalability, and the ease of production, are really kind of standing out there. You're getting good gene editing. What would you say are the limitations of Cas9-CLIPT, or does it have any?
I think some limitations can come just from the gene editing approach and just using Cas9 itself, like the fact that you're using a double strand break to cause and having to resect that and insert the gene. That has typically caused chromosome 14 deletion in probably about 5 to 10% of cells and a lot of these non-viral CAR T products. Some of that has been ameliorated using base editing to knock out the genes, and that has just less of an impact on that. And then you get less chromosomal translocations that way. But you have to do the whole thing with a lentivirus with that.
Yep.
So they're all tools in the toolbox, and they all have pros and cons. Our approach, I think, is good just because you can do the whole thing in one step and you can just axe out viruses completely. And it's easy. It's pretty easy to source a nanoplasmid once they have the sequence and have done the work to sequence it and actually be able to produce it.
Yeah, I mean, I think at a high level, the advantages that you mentioned, Dan, really enable new designs that we can generate, on the computer. We use benchling, but there's AI being integrated into benchling and all types of receptors being, designed and getting that into a human cell. This really, I think, those advantages help there. So I think, generative biopharma type applications for cell therapy, this could help. The limitation, as Dan mentioned, is the genomic impacts of our double-strand break. I think there's ways to mitigate that by doing quality control quite well and analysis of the product. But also, I would say another limitation is really just delivering the template at sufficient quantity into a single cell. And I think we use pretty high concentrations of our clipped template, and we might be at the edge of how much we can get in before a T cell, for instance, dies and senses that we have way too much double-strand DNA floating around. So, 7 kb is the longest template that we've gotten in so far. I would suspect that we can't go up to 20 kb easily this way, such that we have enough template around for homology-directed repair to work at, high frequencies. So there's, I think quite promising techniques out there with these recombinase plus, other types of site-specific integration, bridge editors, et cetera, that you likely need for very long transgenes. But I think there's quite a bit of exciting design space in this 2 kb to 7, 10 kb that you can put in a lot of functionality that would be potentially therapeutic.
No, I agree. I mean, there's also, there's a lot of competition in this space for developing, the new system for whether it be for T cells or for other things. And, as far as I'm concerned, the more the merrier, because if we're all, it takes a village, right?
Absolutely. I mean, I think there's tons of exciting other types of templates, single-strand DNA templates, these recombination, transposase type strategies. As an engineer, the more technologies and techniques that you can de-risk clinically, the more exciting kind of circuits and functionality we can program in. So to me, the future is really bright there.
Right. No, I completely agree. Okay, so to finish, I'd like to go completely off topic here and kind of put you on the spot a little bit. This is a suggestion that Rory, our scientific editor, gave me a couple of podcasts ago, and I really like this one. So this applies to any and all of you, and Dan, especially since you've moved on from the Saha lab. I was wondering, are there any new and interesting projects or developments that you are working on that you're excited about and would be willing to share with us? They don't have to be related to this. Anything that you think is just kind of neat that you're working on? Maybe I'll throw this to Dan first, since he's now in a different lab. Understand if there's nothing that's you're willing to share, but I just figured I'd throw that out there.
Yeah, sure. So on a high level, I met Dr. Soji during my PhD. We collaborated on combining the GD2 CARs with radiation therapy, so like targeted radionuclide therapy, and saw some very interesting benefits when you basically put the two together. They have synergized very well and kind of like have like a 1 + 1 = like 3 kind of additive effect. We've definitely taken some of the approaches that I've used in the Saha lab and tried to extend them to other solid tumors. Quavi is a lot more focused on prostate cancer. He treats that with TRT, and a lot of the work has been targeting like PSMA is a very popular target. So trying to combine that with TRT and kind of push that forward in just different contexts. So it's nice to see how this approach, like Chris has been saying, is very maneuverable, I'd say, and adaptable, and you can use it in different contexts.
Well, great. Good luck with that. And then maybe I'll throw this to Anna. What's in your future?
Yeah, good question. Basically just following up with this at scale production of our CAR T cells and confirming that they are safe at clinical scale for humans and everything. And then also just in the future, maybe looking into multiplex editing. So combining different modalities to kind of improve the functionality of T cells is kind of the space that I'm looking at in the future, maybe.
You're close to graduation, I hope?
Fingers crossed. In my 4th year right now, so hopefully soon.
Well, okay, you got the publication side of it covered already, so that's good.
Thanks.
Yeah, I mean, for me, I'll highlight two things. One is building upon the prior comment of kind of generative AI. We're looking to use receptors that are kind of beyond just the normal targets that and CARs that have been established to bind new antigens on various dysfunctional cells. And it could be not only in cancer, but also in neurodegeneration and other types of disorders. And then the second aspect is looking at analytics during manufacturing. So there's a lot of new data that we can get kind of in line or near at line during these manufacturing runs, and especially with some of the capabilities that Solaris has that is in a fully automated system. Can we increase our efficiencies better to really make products that are more potent, such that we can lower dose and make these types of cell therapies more affordable? Those are potentially places in terms of getting data and doing machine learning or other analytics to really make these manufacturing runs more efficient.
Well, that's fantastic. Well, I want to thank you all, Chris, Anna, and Dan, for joining us today and, talking about your work and kind of, the past, the present, and the future. It's been very helpful. And this is obviously a very, a field that's already big, which I feel is going to become, much, much bigger as we, get better at developing these immune cell therapies that seem to be working pretty well in the clinic. So thank you for that.